Hello, grade 11 biology class. Since there are a few students that are not with us here in Vientiane, or they are in Vientiane but not at school, I am going to record a couple of lessons for them in order to help with their studies. Also, I've put my face in the corner there. I hope it's not going to block too much of the presentation as I go through it. But you would be advised anyhow to go through the presentation yourself also, because if I were to talk too much with this presentation, because it has, look here, 38 slides, so even if I would talk only for a minute per slide, then it will already be 40 minutes long, and I don't want to make these presentations, video presentations, too long, because they will become too boring also. So, what we start off with is the cell, and just an introduction, which this subunit talks mostly about stem cells, and starts off with first a listing of the functions of a life that are performed by each individual cell. Now, the first one is nutrition, feeding. Basically, there is a few different ways how you can, how an organism or a cell can feed itself. And we are very reliant on so-called autotrophs, plants, but also algae, for instance, unicellular organisms that are able to photosynthesize. Yeah, I'm sure you're well familiar with photosynthesis to the extent that you know that it's a process in which inorganic substances like water and carbon dioxide are turned into organic substances like glucose, for instance. Heterotrophs are dependent on that process because heterotrophs and herbivores initially are the ones that eat the results of all that work that the autotrophs do by eating plants. And then other heterotrophs that are higher up on the food chain, they will eat those herbivores, for instance. So we call them. These are also known as producers because they produce organic material, and heterotrophs are known as consumers. And then you got decomposers that close the cycle again, the nutrient cycle, by turning all the organic materials back into inorganic materials that will go back into the pool from which the autotrophs make their organic materials. So there's a nice cycle there happening also. We'll come back to this in more detail in the ecology units. Metabolism, the second function of life. The metabolism refers to all of the reactions that take place in your cells, in all your bodily cells. In every cell, basically, there's an enormous amount of reactions taking place, and we'll look into more depth into some of them, like for instance photosynthesis, respiration, which is the creation of ATP, the energy molecule in your cells, but also protein synthesis, DNA replication, transcription. But there's much more happening also. Your body's cells are able to take proteins and turn them into carbohydrates, and carbohydrates into fats, and vice versa, as an example also. So there's an enormous amount of metabolism reactions, chemical reactions happening in your body. And they also produce waste products, and we'll come back to that in a second when we talk about excretion. First, growth. Growth is not a function of life. If you are, you're not, but if you were unicellular, then what you can do is you just split yourself into two. So this little organism here that's known as a paramecium, but the same thing applies to bacteria. They are able to simply split up into two, and we call that process binary fission. If I go here, then the pop up, but so you can see that word there, binary fission. That's the way that many unicellular organisms reproduce and hence grow in numbers. Multicellular organisms, like we are able to divide our cells, cell division, and that is called mitosis also. We'll speak more in detail about mitosis later on. Mitosis is a process in which cells are copied, basically so you have a mother cell, like here for instance, fertilized egg cell, and that becomes two cells, and those two cells are genetically identical to the mother cell. So response to the environment, to triggers in the environment. obviously, we have all sorts of sensors built into our body, audio, visual, etc. All our senses that make us respond to the environment. But less obviously, perhaps, plants also respond to the environment. Phototropism here, for instance, that's the ability of plants to grow towards light. Plants don't have a nervous system as such as we have, but they've got plant hormones that regulate the direction of growth. For instance, if you are a HL student, we'll do plant biology at some point, and you'll be learning more about those particular plant hormones. Phototropism. There's also unicellular organisms. This one here is called Euglena. That is a protozoa, unicellular organism that has an eye spot actually there. It has light-sensitive molecules in its body. So it's not actually an eye, of course. This is one cell only, and that eye spot is able to detect light. So it can actually, you think about it, what would Euglena, who has chloroplasts also, which means that it can photosynthesize, it's that sounds like algae, what would it like to do? Move towards the light or away from the light? You can figure that out, I'm sure. And it moves by means of a flagellum there. It has a whip-like tail. Another response, Venus flytrap, gets triggered by movement on its. These are leaves actually. They're modified leaves, and on top of those leaves you'll have a couple of spikes. And those spikes, when they get touched by a fly, for instance, that lands on the Venus flytrap, after 2 touches, it closes its leaves. So it can actually count in a way. It builds up chemicals as soon as one of these spikes is touched, and if there's enough chemicals, so on the second touch, then there's so much chemicals that it actually responds to by closing. Chemotaxis, that's the ability of certain bacteria to detect molecules in their surroundings, for instance, molecules that were left by a prey. And these all organisms, all cells have receptors on their cell membrane, and these receptors are able to pick up those signals and then actually follow them around. Okay, so that's response. Excretion, I mentioned it already a little bit. Excretion, getting rid of waste products of metabolism, like for instance breathing out, getting rid of CO2, sweating, peeing, for instance, also because the kidney is able to filter out your blood. Egestion, which is a nice word for pooing, is not excretion, by the way. That is often confusing for students because once you eat something, it doesn't really become part of your body and doesn't enter your cells until it's absorbed in the small intestine. If it's not absorbed, you are getting rid of it on the toilet, let's say, and that's egestion. So, but what happens there, number 2, let's say, has never been part of your body. So, excretion refers to the processes that you see here, not egestion. Homeostasis, that's the ability of your body and ability that all multicellular organisms in particular have to respond to the environment and keep their internal environment at a certain stable level. For instance, keep maintaining your temperature in your body. And there's all sorts of mechanisms that are built in. Sweating, vasodilation, that's the increase in diameter of your blood vessels, for instance, are a response to an increase of temperature. Because if your the blood vessels in your arm increase in size and come closer to the surface, you will lose heat more readily. So there is a negative feedback, meaning there's a response to an increase in temperature to bring the temperature down, and there's a response when the temperature drops to bring the temperature back up again. And that's called negative feedback. Same thing sort of happens with blood sugar levels. There is a hormone called insulin that you must have heard of, I'm sure. There's also one called glucagon, and they do opposite things, but they will bring, insulin will bring blood sugar levels down in a certain way. We'll come back to the details of that later on. There is also, wrong direction. There's also such a thing as osmoregulation. to make sure that your body maintains the proper amounts of water. And that's mostly the work that your kidney does for you. Paramecium, you see paramecium here again, has a so-called vacuole that can contract, a contractile vacuole. So it's actually collecting water in that vacuole, and when it contracts, it's actually squirting out water from its body also. So even unicellular organisms have a mechanism for this. Now, reproduction, that's a very important part of life. What's the point of living otherwise, you might say, unless you pass your genes on to the next generation? This can happen sexually and asexually. It can asexual reproduction in plants is not uncommon at all. This is an example of strawberry plants that can have these so-called runners. So one plant can run away from the mother plant, let's say, and form new roots and grow again. I'm sure you're familiar with bamboo does the same thing. Bamboo has underground runners. More examples of palm-like plants that you might find in your own garden that do the same thing. Here again, Paramecium can also sexually reproduce, and that is by exchanging DNA in a process called conjugation. Actually, sorry, I have to rectify myself. It's not actually a sexual reproduction, but they do exchange DNA there also. Now, there's some peculiarities in nature that are extreme things. Parthenogenesis, that's the ability of certain mammals to also reproduce asexually. There's a nice, interesting article here. Maybe you want to click on that because these are the stories that make biology so interesting. So here there is a sawfish in Florida that has been seen in the wild with offspring that carry only its own DNA and no male DNA. So it's an interesting article there to read for you. And the earthworms here, they are hermaphrodites. It's also maybe interesting. They line up with each other in what you might call a 69 position and exchange sperm cells and egg cells. They're also sperm cells mostly. So both get fertilized in that process. Now, this is the conjugation here, and also the whole idea that all of these functions of life are also performed by one particular cell. So we have here a connecting tube, you might say, that connects both of these bacteria together, and that's known as a sex pilus. Now, here in the bottom you see 1 micrometer. This here reads as 1 micrometer, and the question is, what is the enlargement of this image? You need to be able to do those calculations. There's a bit of mathematics there. Now, the indicator here sort of means that if this is 1 micrometer, that's this bacterium here is about 2 micrometers long. But how much has this been enlarged to see it to this extent? Now, a micrometer is 1,000th of a millimeter. So if you think of a millimeter, and a millimeter, you know from your rulers, so you have a centimeter divided by 10, and you get to millimeters, and a millimeter divided by 1,000 gives you a micrometer. So that means there are 10,000 micrometers. Think about it, 10,000 micrometers in a centimeter. Now this here, you have to measure it exactly, but this looks to me like roughly a centimeter. So that means that the magnification here is 10,000. 10,000 times. So this organism is 10,000 times as small as you see it here on the screen, which is pretty small. You have to be able to give some examples of, for instance, Paramecium and another protozoa. There is a good clip for that here. So if you watch this particular clip, somebody has made the effort to explain all of the functions of life for Paramecium and Chlamydomonas. So I'm not going to show you that video now, obviously, but I would
Paramecium.
note of that. So watch that video, and here also you can see some indications here of Paramecium with the functions of life. So make those notes. Now, then we come to cells. A little bit about what's the origin of cells. We will look in the next unit into prokaryotic cells versus eukaryotic cells. How old are prokaryotic cells? Because they were the first one on the planet. I wrote here the earliest cells date back a staggering 3.8 billion years, roughly, because we're not quite sure yet. It could also be 4.1 billion years, or maybe even a little bit older. Earth itself is roughly 4.6 billion years old. So the age of life on this planet is not too far off from the age of the planet itself. So the Earth had to cool down first for life to become possible. So yeah, it may have been 800 million years. It may have been even a little bit less. Then life came about in the form of unicellular prokaryotic, very simple cells. It would be good to click on the link there to go to this website, which is by the University of Utah. They have a lot of, and it's University of Utah, but it's very accessible stuff. They got a lot there on the evolution and genetics of cells. And they also talk here about the endosymbiotic theory. You have to be in the know of what the endosymbiotic theory actually represents, what it means. We'll talk a little bit more about it in our presentation also, but you can also already read up a little bit here about it. So endosymbiotic theory explains how eukaryotic cells came about, and that was basically due to the symbiosis between different prokaryotic cells. Now, I'll leave it at that for you to read. And this here is Mr. Spock asking, where did these cells come from? Now, there's two basic ideas. One very accepted idea is the primordial soup hypothesis, which refers to the whole notion that the first forms of life were indeed found in deep sea vents, where there was a lot of hot gases bubbling up from the magma. That's liquid magma that we find in the core of the Earth, bubbling up there. And magma contains a lot of minerals, and those minerals were inorganic, but they could have been energy sources for early life forms. So the primordial soup hypothesis is quite well accepted. There is also the idea that maybe life was brought to this planet, didn't actually start here, but it started somewhere else, and it was brought to us by comets, meteorites, and there have been meteorites found with amino acids on them. So organic molecules do exist out there in space. We've not found actual cellular life out there in space. So both theories are valid. Basically, the idea is that you're not alone in this universe, and there's two typical notions. I found this one very interesting to think about. Either we are alone, so you have to imagine the immense vastness of this universe. We're talking about we are living in a solar system that's part of the Milky Way as a galaxy. Now there are billions of galaxies in the universe, and each galaxy consists of billions of planets. So imagine that there's only one of those billions of planets that contains life. That's a pretty scary notion. But if there's life out there also, that's also a terrifying sort of idea. It leads to a lot of interesting science fiction movies, not scientific movies, science fiction movies. Discovery of cells, you need to know a little bit about cell theory and about the people that were the founders of this cell theory. First of all, Antony van Leeuwenhoek, that is the guy who first described unicellular life, and he was a merchant in textiles, and he needed magnifying glasses to check the quality of his textiles, and he developed he became very good at making lenses. And he also developed this device here. There's a lens there where my red dot is now. That lens was able to magnify up to about 500 times, which is pretty good, because even the microscopes that we use in class can only magnify up to 400 times. And he made drawings there, and he sent those drawings to the Royal Society in London. to say, I found something in the waters in Holland, because he took water samples from the lakes nearby his house, and this actually is his sperm, not from the samples of water, but he sent those drawings to London to this fella here, Robert Hooke, and Robert Hooke was receiving letters from Leeuwenhoek. The names are a bit similar, but different. And he was a bit annoyed because he was also developing microscopes, but he didn't see those microscopes. He didn't see those microbes that van Leeuwenhoek saw. And his conclusion was basically that Holland must be a very dirty country that these little organisms existed. In the end, his microscopes improved, and he also saw cells, and he started drawing them up. And he was the one responsible for the term cell, because it reminded him of cells in honeycombs. Now, then Robert Brown, he coined the term nucleus. And I don't know if you are familiar with Brownian movement. That's the movement of all molecules given a certain temperature. That's also one of his findings. Then there were two gentlemen from Germany, Schwann and Schleiden, and they came up with the first bit of the cell theory. That is that all organisms consist of cells, either one cell or more cells. That no organisms have been found, let's say, that don't consist of cells. So they came up with that part. And then there was a fella also from Germany, Virchow, and he said something one morning, much to the delight of people around him, I'm sure. He said, omnis cellula e cellula. And that means, omnis means all, cellula refers to cells, a means from, so this means all cells are from cells. And that is the basis of cell theory. So cell theory, you need to be able to reproduce this knowledge. All living organisms are composed of one or more cells. The cell is the most basic unit of life, and all cells arise from pre-existing living cells. Yeah, so take note of that. There are some exceptions. That's the interesting thing about biology, that there's always a little exception to the rule in general. Muscle cells, for instance, are striated muscle cells are that's the ones that run across your run around your bones and allow you to move. They have more nuclei per cell, so they actually don't follow that whole idea that a cell is the smallest unit of life, because they are combined cells together. And we see that also in plant life. There are seaweeds that consist of big cells, basically very big cells with many nuclei. So their smallest unit is not so much the cell, but something bigger. Now, then moving on to something called the surface area to volume ratio. The whole idea is, why is an elephant, for instance, a big organism, why does it have more cells? Why can't it just be one big cell? And the reason for that is that there is a restriction as to how big cells can become without getting into trouble. The trouble being that if a cell becomes too large, indicated, for instance, by this huge cube here, with like a Rubik cube, then the distance from the middle of the cell to the cell membrane would be relatively large. The distance would be long, and then the cell would not be able to take in oxygen quickly enough and to get rid of waste products quickly enough. So it would run out of oxygen and build up too many waste products. Now, the calculation and mathematics behind it is that if we calculate the volume and the surface area of the cubes here, that you'll see that if we divide, and you can check the numbers. I hope it's all correct, because I took this image from somewhere else. Yeah, 4 times 624. Yeah. Okay, it's fine. So here, if you divide the surface area by the volume, you'll find that the smaller the cube becomes, the larger the surface area to volume becomes. And if the surface area to volume ratio becomes too small, then the cell becomes basically too big, and it has an issue with that, then the ratio gets too small, limits the size of cells, and it's explained here also. So I said cell can't get rid of its waste products anymore, including also heat that it produces. So it becomes too warm, basically. Now, the advantage of having to split up into a multicellular organism, so having more cells, is that our cells are able to differentiate into different types of cells. So we start off as a singular cell, so that is a zygote, the fertilized egg cell that was formed by the fusion of an egg cell and a sperm cell. And that one cell that you once were has started to split up into 2 cells, 4 cells, etc. And then in the process of your embryonic development, started to differentiate into different cell types. So now in your body, you have about 200 different cell types, which is wonderful because they're all responsible for a different task that they can perform. And that's why we are quite versatile organisms compared to, for instance, bacteria that are relatively simple or other unicellular organisms. Okay. The advantage of having so many cells also, like for instance, think about your brain and compare that maybe to the internet also, where we have a connection, a network between different terminals, laptops and servers and all that, is that you might consider the internet an emergent property of that, the cloud, as we call it also. So your consciousness, the whole idea that you can think and that you have a certain personality, for instance, is an emergent property from the simple fact that all your neurons, all your brain cells are connected together. And then from there are able to do something bigger than themselves, you might say, that a single individual nerve cell couldn't do. So there's an idea of synergy and emergent property. So consciousness is a good example of that. Now, a couple of, I mentioned zygote already. I don't know if you're familiar with that term, but that's the first single cell. That was you. A zygote is totipotent, can become any type of cell still. And in the course of your embryonic development, it starts to differentiate into different types of cells, although embryonic stem cells, are still called pluripotent. They have also the ability to develop into different other cells, not as much as the zygote, though. Somatic stem cells in your body, somatic means non-sex cells, they are multipotent and they can differentiate into a few different types of cells. Think, for instance, of your bone marrow. In your bone marrow, in your bones, there are stem cells that can differentiate, that can be copied into either white blood cells or red blood cells, different types of blood cells. Now, this is a simple exponential growth there that you see, 2, 4, 8, 16, 32 cells. And as you grow, this is a typical development here, you will see that those cells will start to differentiate until they become specialized at this stage here, when they are actually lung cells, thyroid cells. That's thyroid is in your thyroid glands and your pancreas, etc. They have so many different functions and structures that we call them specialized. Now, one big question here, maybe I can talk about it a little bit as well later on. All of your bloody cells have the same genetic information, because in mitosis, what you get is that the daughter cells are genetically completely identical. So how is it that a muscle cell knows it has to become a muscle cell, and a nerve cell a nerve cell? So it must use different genes in the expression as it grows. And how is that achieved? Now, one of the things is induction. Induction is the whole ability of one cell to influence a next cell next to it, and that is done by releasing certain triggers, you might say, certain molecules that can be picked up by receptors. Again, that word receptor on the membranes of cells, and those receptors will then possibly do something with that signal that they receive, and they can actually send information to the nucleus to do something. We see a similar thing with hormones. If a hormone is sent around your body, then the cell receptors will pick up that hormone and send a signal to the nucleus to start producing certain proteins, for instance. So that's induction. When cells are copied, as mitosis occurs, the division can also be so-called asymmetric, which means that although the nuclei, the genetic instructions are the same, the cytoplasm doesn't have to be the same. So stem cell might be, when it copies here, for instance, you might get one stem cell that remains, for instance, in your bone marrow, and a copy that can actually differentiate into a different kind of cell. Now, this here is also an interesting story. This is DNA, and your DNA is wound up around histones. In its most condensed form here, you'll see that it's completely packed, wrapped up around those proteins. The blue parts, histones, are proteins. And if it's completely wound up like this, super condensed, as you say, super coiled, then the genes on the DNA there will not be expressed. In contrast to when it's opened up like this. So if there would be a gene here on this particular DNA, now it can be expressed. If it's completely coiled up like this, then it will not be expressed. And the cell has a mechanism to determine the amount of coiling, of condensing. So that is probably most likely also a mechanism by which cells determine how certain cells become nerve cells and how other cells become muscle cells, etc. Then there is such a thing as gene therapy. Embryonic stem cells are used in gene therapy. There's a good clip here. Again, I urge you to watch that. One of the examples is bone marrow transplants, where in the case of leukemia, you might have heard of that. That's sort of blood cancer, where your white blood cells, also known as leukocytes, leukocytes is a name for a white blood cell. The white blood cells don't function properly because the stem cells in leukemia patients, have been mutated, altered, so that they don't function properly. Now, what happens then is that a healthy donor with a similar blood type, most likely, will donate bone marrow, and the patient will have its bone marrow removed, and then the bone marrow will be the healthy bone marrow will be injected into the patient's body, and that's if it occurs without any issues, then that healthy bone marrow will then start to produce healthy blood cells, white blood cells for the patient. Okay. There's another disease. This is mentioned in the syllabus, something that you have to be familiar with. Stargardt's macular dystrophy, which is a disease that leads to loss of vision, eventually to blindness. That is also due to a mutation in stem cells in your retina. So there are stem cells that are able to generate stem cells for your retina. And you can be injected with new stem cells. If you have that particular disease, then it's possible to insert stem cells there, and they will replace the mutated stem cells. There's an interesting question here. Are there any ethical considerations when embryonic stem cells are used? And you might want to give that some notion also, because what is the source of stem cells used in these kind of treatments? And often it was embryonic stem cells. So where are they from? Now, I'll leave that up to you to figure it out. Here's another bit of a side story. I put it in there because it was interesting, I thought. Meat production. So there is a huge issue with traditional meat production, intense farming, etc. Comes with a lot of ecological impacts. So people like to eat still. In an effort to push towards eating more ecologically, environmentally friendly meat, stem cell hamburgers. So you could grow meat in the lab, basically. issue there is still is years away, I think, from being economically viable. Issue is that it also needs to have a certain bite to it, and getting those fibers in the stem cell hamburger is a difficulty, I believe. Now, blood made in the lab is also interesting. Read the article, I would say. This article is from the Guardian. If you get, there might be a message from the Guardian that you need to log in for free, though. Quickly, you can sign in with Google, the Guardian. And this is an interesting article. This whole development of stem cell blood, it was based actually on a desire of the American Army to create a machine that could make blood for war zones. Because there's a lot of blood transfusions happening generally, there's a lot of people that need blood in a war zone situation. And if you could create type O blood, which can be universally given to patients in the field, then that would be a great way forward. So read this article. Also, I won't elaborate too much. Now, this website here is also linked on my. If you go to my website, there are here stem cells, University of Utah. I would urge you to have a look around there. Also, and there are some very accessible go-go stem cells. Is a very nice activity to do. Very simple, relatively simple, very accessible. Last thing that I want to mention is induced pluripotent stem cells. That's the latest development where we are artificially able to reverse the specialization and differentiation of cells, and that is earlier that was always thought. Sorry, my English. That was always thought to be impossible, but a mechanism has been found that we can actually reverse that process. This here is a clip with Dr. Yamanaka, who was the inventor of this technique, if you're interested to hear about that. And using those techniques, now we can start to grow maybe organs. So we can take one of your body cells and reverse that, even just a skinned cell, and reverse that into a pluripotent cell and grow any other organ that you might be lacking or that's not working properly. Heart tissue, for instance. They've done it already with teeth in mice also. These are all links to articles that you can read. Even now, they have been able, and this is already from 2014, if you click on it, for this interesting already, that so the research, usually they generate blank slate stem cells, it says here, but now they have been able to flip a genetic switch in the DNA and convert from one cell type into a different cell type, just like that. So this would be wonderful news for possible future developments. I did see something interesting. I thought it was interesting to point out that this kind of technology will take years and years and years before it can be developed safely. Oh, here it was. The technique so far only tested on mice. It might be another 10 years here before such a treatment is shown to be effective. So we read a lot about what's possible with gene therapy and stem cell research, but things that happen in the lab with mice are not necessarily things that are still are going to be able readily anytime soon with human beings also, because there's an ethical issue to in terms of how you're going to actually try this out. You can't just take a human being and give it a go. There's of course ethical considerations. Now, there's also an interesting development here. Infusions of young blood may reverse effects of aging, studies suggest. It's a suggestive study here. What they've done is they have conjugated mice together. I think there's a clip here that indicates how this is done. Basically, they connect blood streams of two different mice. I'm not sure if this video actually shows it. I thought it did, but no. Let's see if there's a picture of it. No, sorry. Okay. Now, what they do is they connect the bloodstream of two different mice, one old mouse, one young mouse, and then they find that the young mouse gets a little bit older, and the older mouse rejuvenates a bit, and you can measure that by looking at the proteins that flow through the bloodstream. Interesting, very interesting idea, leading to all sorts of sinister scenarios whereby you can imagine if you infuse yourself with embryonic blood, for instance, that you might become younger, and even as an interesting link to Dracula. Yeah, the whole idea that vampires stay young by what do? They suck the blood of virgins. Now, if there's a nice link there between the two ideas, but there is going to be maybe a scientific background to this. Okay, I'm going to leave it at this. I'm sure this video is already pretty long as it is. So go through the PowerPoint yourself, this presentation, take some notes. Also use Cognity where necessary. And I'll be sending you some assignments to do. Okay. All right. Good luck. Let me know where you are.